Good morning, students. Today's topic is PCR technology. Okay. Already we have completed 50% portion of this chapter. Already we have completed. We have discussed what is biotechnology, the principles of biotechnology, the raw materials of RDT technique, the process of RDT technique, and different types of the technology which we can use for the recombinant DNA process to from the recombinant DNA, the gel electrophoresis process. Okay, already we have discussed all those things. So today we are going to discuss about the PCR. Okay, so what is PCR? PCR, the full form of PCR is the polymerase chain reaction. The full form of PCR is polymerase chain reaction. Okay, so what is the benefits of PCR? What is the usefulness of the PCR? That this is a technique that takes specific sequences of DNA of a small amount and amplifies it to be used for further testing in vitro process. Okay, so what is the meaning of the PCR? PCR means that through this technique, through this polymerase chain reaction, we get high amount of or big amount of the desired DNA sequence from a very small amount of DNA. Okay, so this is the basic things of the PCR. So what is the purpose of PCR? To amplify. Amplify means the to getting a large amount of thing. Okay, it means the replicate. To amplify a lot of double-stranded DNA molecules with the same size and sequence by enzymatic method and cycling condition. So what does it mean? It means to amplify a lot of double-stranded DNA molecules with same size and sequence by enzymatic method and cycling condition. So what is the meaning of this? That the through this technique, through this PCR process, we can get a large amount of fragments of DNA molecules from a single fragment of a DNA molecule. Okay, and we get same identical size and sequence of these DNA molecules after the PCR process by the enzymatic method and cycling condition. So we can say in this way that the PCR is also one type of enzymatic method. Okay, basic requirements for PCR technique. DNA sequence of target region must be known. Okay, after when we are using this PCR method, when we get to know that this sequence, we need this sequence, we need this particular sequence of DNA. This sequence is our desired sequence. After that, after knowing this desired sequence, then what happen? Then we can add the primers, typically 20 to 30 bases in size. This can be readily produced by commercial companies, can also be prepared using a DNA synthesizer. Next is a thermostable DNA polymerase, example Taq polymerase, okay, which is not inactivated by heating to 95 degrees centigrade. So this Taq polymerase is coming from the bacteria Thermus aquaticus. Okay, Taq polymerase, we get Taq polymerase from the bacteria Thermus aquaticus. Okay. DNA, next point is the DNA thermocycler. It's a machine which can be a program to carry out heating and cooling of samples over a number of cycles. Okay. So what is the condition or what is the procedure of PCR? The first is the denaturation of DS DNA template. DS means the double-stranded DNA template. Next one, annealing of primers. Third one, extension of double-stranded DNA molecules. First one is the denaturation of double-stranded DNA template. Second one, annealing of primers. Third one, extension of double-stranded DNA molecules. Clear? Okay. So next is the denaturation. What is the meaning of the denaturation? Denaturation means the when the double-stranded DNA is transformed to the single-stranded DNA. That's called the denaturation method. So this DNA is always the thermosensible genetic material. Okay, what is the meaning of this? The temperature sensible. It means in the high temperature, the double-stranded DNA breaks down into the single-stranded DNA. Okay, so the temperature is 92 to 94 degrees centigrade. Okay, here you must know one thing, that this temperature parameter, this temperature range, the range of temperature is very important. Okay, during the time of your exam, when you will write this answer, you have to remember the exact temperature. Okay, because this process is temperature sensitive process. If you write the wrong temperature, the range of wrong temperature, then your answer will be incorrect, your whole answer will be incorrect. Clear? So during the time of your exam, please remember one thing, that you have to write a proper range of temperature. You have to write this particular temperature. You cannot write that 93 degrees centigrade. Okay, you have to write 92 to 94 degrees centigrade. Okay, so the temperature is 92 to 94 degrees centigrade. The double stranded DNA melts into single stranded DNA. Okay, after that, what is the meaning of this? Look, this is the double stranded DNA, 5 prime to 3 prime, 3 prime to 5 prime. This is the double stranded DNA. After the heat therapy, the heat is 92 degrees centigrade. So look at the temperature. After the heat therapy, then it transforms to the, it breaks down to single stranded DNA. One strand, another strand. Okay, 5 prime to 3 prime, 3 prime to 5 prime. That's the process is called the denaturation process. This is the process is called the denaturation process. Okay, next one is the annealing. What is the meaning of the annealing? That the temperature is here, the temperature, we need the temperature 50 to 70 degrees centigrade approximately. 50 to 70 degrees centigrade, and you have to write this exact range of temperature. You have to write this exact range of temperature, otherwise your answer will be incorrect. I am repeating again and again, because this whole process is the temperature sensitive process. This whole process is temperature sensitive process, so that's why please remember the exact range of temperature. So what is the thing of the, what is the main phenomenon which has happened in the annealing? That the temperature range is approximately 50 to 70 degrees centigrade, and then dependent on the melting temperature of the expected duplex. Okay, melting temperature of expected duplex means the, this is duplex means the double stranded, this is the melting temperature. Okay, so based on this melting temperature, this annealing temperature is vary. So after the heating treatment, the primers bind to their complementary sequence. This is the primer. Okay, next one is the extension. Here that we need temperature approximately 72 degrees centigrade. Why? Because in this temperature range, in this 72 degrees centigrade, what happened? The Taq polymerase become activated. Okay, Taq polymerase is just like the DNA polymerase. In the case when this DNA polymerase, we'll see, actually we have seen this DNA polymerase activity in our DNA replication portion. Okay, so for each and every this type of process, we need one polymerase. So here Taq polymerase plays a pivotal role to maintain this extension process. Okay, so DNA polymerase binds to the annealed primers and extends DNA at the 3 prime end of the chain. So DNA polymerase binds to the annealed primers and extends DNA at the 3 prime end of the chain. Okay, so this is the whole process of the PCR technique. This is the whole process of PCR technique. Clear? Okay, next is the DNA fingerprinting. What is DNA fingerprinting? So DNA fingerprinting is a way to identify a certain individual rather than a simple identifying a species of a particular trait. Because we all know that the DNA fingerprinting process, this process or this technology is quite familiar to all of us. We all know what is DNA fingerprinting, and we know one thing, that through the DNA fingerprinting we can get the criminal. Okay, this is our basic idea. So you should know what is the, how this DNA fingerprinting works. Okay, so DNA fingerprinting is one kind of technology to identify a certain individual rather than a simple identifying a species of a particular trait. Okay, a technique used by scientists to distinguish between the individuals of the same species using only samples of their DNA. Okay, so from the samples of the DNA, from the one samples of DNA, we can know, or we will know that the criminal. We will know about the criminal. So biological sample used for DNA fingerprinting is blood, hair, saliva, body tissue. DNA samples have been obtained from the vaginal cells transferred to the outside of the condom. Okay. Next is the steps of DNA fingerprinting. There are normally 8 steps for DNA fingerprinting. These steps are very important for your exam. These steps are very important for your exam. So please go through it properly, because this question is very important for your 3 to 5 marks question. Okay. The first step is the isolation of DNA. This is the first step, the isolation of DNA. DNA must be recovered from the cells or tissue. Only a small amount of blood, hair or skin is needed to isolate the DNA. So DNA must be recovered from cells or tissues. Only a small amount of blood, hair or skin is needed to isolate the DNA. After that, this is the step 2. The digestion of DNA by restriction endonucleases. The DNA is cut into fragments using restriction enzyme. After the cutting, each restriction enzyme cut DNA at a specific base sequence. Okay. Look, this is the specific sequence. Okay. Either staggered cut or blunt cut. Clear? Next one. The sections of DNA that are cut out are called the restriction fragments. This yields thousands of restriction fragments of all different sizes, because the base sequences being cut may be far apart or close together. This is either the long fragment, it will be either the long fragment or the short fragment. Okay. Commonly used restriction enzymes are the Haemophilus 3, LE1, etc. Restriction mixture is incubated overnight at 37 degrees centigrade. Next is step 3, electrophoretic separation of different fragments. Okay, like the electrophoresis. So fragments are separated on the basis of size using a process called the gel electrophoresis. DNA fragments are injected into wells and an electric current is applied along the gel. This is the anode, this is the cathode, just similar the gel electrophoresis method. Okay. DNA is negatively charged, so it's attract to the positive end of the gel. It means the anode. Okay, this is the, as because the DNA fragment, the charge of DNA fragment is negative, so that's why it moves towards the positive end. The shorter DNA fragments move faster than the longer fragment. DNA is separated on the basis of size, just similar to the gel electrophoresis method. Okay. Next is the step 4, transfer of DNA on nylon or nitrocellulose membrane. The DNA fragments are transferred to a nylon sheet by placing the sheet on the gel and soaking them overnight by the process southern blotting. Okay, so this is the process. This is the agarose gel, this is the nylon membrane, this is the Whiteman paper, this is the rubber gasket, and this is the porous mat. Okay, this is the vacuum blotter. Okay. Step 5, probe labeling. Adding radioactive or colored probes. Why? Actually, what is the use of this probe? To identify a specific segment. Okay, to identify the specific segment of DNA to the nylon sheet, which is complementary to target sequences. Each probe only sticks to one or two specific places of the sheet. Okay. Next is the hybridization. Label probe DNA should be hybridized with the complementary sequences located on nylon membrane for the detection of position of latter. Membrane is used to remove non-specific binding and clearing of the background. Okay, this is the nylon sheet. 7th, autoradiography. To detect the sequence in genome bound with the hybridized radioactive probe on membrane, this technique involves the alignment of the hybridized membrane with the X-ray film. The X-ray film alters its development, shows the multiple number of bands. Look, different number of bands are there. And the last one is the interpretation of band patterns. and the analysis of band patterns of different individuals, comparison of position of bands, and computer software are also available for the analysis of DNA fingerprints. Clear? So this is the basic techniques of the DNA fingerprinting method. I am repeating again. The first one is the first you should isolate the DNA from the cell. After that, this DNA become digested by the restriction endonuclease. Third, the electrophoretic separation of different fragments. Okay. Fourth, transfer of DNA on the nitrocellulose membrane. Fifth, that the probing or probe labeling. Sixth, hybridization. Seventh, autoradiography, and then interpretation of the band pattern. So through the interpretation of band patterns, you will get to know that he or she is a criminal. For this, so that's why this DNA fingerprinting process or method is very useful to detect the criminal. Okay. So what is the uses of DNA fingerprinting method? That first, diagnosis of inherited disorder. Second, it helps diagnose disorder in both prenatal and newborn babies. Third, disorders may include cystic fibrosis, hemophilia, Hutchinson disease, all those things. And the last, the main thing for the to detect the criminal. Okay. Forensic science is the use of scientific knowledge in the legal situation. The DNA profile of each individual is highly specific. Okay. So, and you will get to know about the twins or any problems are there between the twins. Then you will get to know through this DNA fingerprinting method. Okay. And the next is the northern and southern blotting. So this northern blotting is a technique used for the study of gene expression. These two are very important for your 2 marks question. This northern blotting and the southern blotting. Okay. Northern blotting is a technique used for the study of gene expression. It's done by detection of a particular RNA. mRNA is generally represented as 5% of the overall RNA sequence. This method reveals the identity, number, activity, and size of the particular gene. So when you need to know about the RNA sequence, or you have to do the isolate the mRNA, you have to isolate the RNA, then you can use this northern blotting technique. And in case of the southern blotting is a laboratory technique used to detect a specific DNA sequence, okay, in a blood or tissue sample. A restriction enzyme is used to cut a sample of DNA into fragments that are separated using gel electrophoresis, and after that these DNA fragments are transferred to the gel base of the membrane. So this is the two process. Through the northern blotting, you will get to know about the gene expression, actually particularly the RNA sequence, and through the southern blotting you will get to know the specific DNA sequence. Okay. So this is the bioreactor, the last process or the last raw material of the RDT technique, the bioreactor. Bioreactor is an apparatus for culturing organisms like algae, fungi, bacteria, animal cell or plant cells under controlled condition. Okay. It's used in industrial process to produce the pharmaceuticals, vaccines or antibodies. In bioreactor, raw materials are biologically converted to the specific products. Okay. The most commonly used bioreactor as stirred tank reactor. Look, this is the structure of the bioreactor. This is the stirred tank reactor, and this bioreactor has some specific function. An agitator or stir system. Look, second, an oxygen delivery system. One, air outlet. This means the oxygen air inlet and air outlet. It means the oxygen delivery system. A foam control system. This is one foam control system. This inoculum, this is the foam control system. A temperature control system. Okay. Next one is the pH control system, and the last one is the sample ports. Okay. So this is the overviews of the bioreactor, and the last one is the downstreaming process. The separation purification of the products obtained from the recombinant DNA technique is called the downstream process. Okay. The product has to be formulated with suitable preservatives. and the clinical trials and strict quality control testing is also conducted before marketing of the product. Okay, so what is our basic need from the RDT technique? To get a desirable product. Okay, to get a desirable product, this is the basic need, na? So through the downstream process, we will get our desirable products. Okay, and by which process? That the separation, purification, and of the products obtained from the recombinant DNA technique is called the downstreaming process. Okay, so all we have completed this chapter. Okay, this chapter is completed. So if you have any queries, ask me. Okay, go through my video, and if any help is required, if any help is needed, you can see the slides here. Okay, you can see the slides here. There you will get different kinds of the biological PPT. Just search your main criteria, what you want to know. There you will get the PPT presentation on this topic. Okay, so go through it properly, and if you have any queries, ask me. Okay, thank you.